Patenting and affairs. After investing enormous amounts of time and money on research and development, a pharmaceutical company needs to reap the benefit of all its hard work. To do so, it needs to have the exclusive rights to sell and manufacture its products for a reasonable period of time, and at a price which will not only recoup its costs, but will generate sufficient profits for further research and development. Patents allow companies the exclusive right to the use and profits of a novel pharmaceutical for a limited term. Patents in most countries run for 20 years after the date of filing. Each country has its own patents, so the company has to first decide in which countries it is going to market its new drug and then file the relevant patents. Once a patent has been filed, the patent authorities must decide whether the claims are novel and whether they satisfy the necessary requirements to get the patent body. There is a universal golden rule. Pharmaceutical companies only reveal their work after the structures involved have been safely patented. Patents are filed as soon as a novel agent or series of agents is found to have significant activity. This prevents a rival company from making a close analog of the specific structure and selling it in competition. Most patents are designed to cover a series of compounds belonging to the particular structural class rather than one specific structure. Nowadays, most of the drugs reaching the market are either chiral or pure enantiomers. Chiral switching relates mostly to racemic drugs that have been on the market for several years and are approaching the end of their patent life. By switching to the pure enantiomer, companies can argue that it is a new invention and take out a new patent. However, they have to prove that the pure enantiomer is an improvement on the original racemate, and that they could not have reasonably have been expected to know that when the racemate was originally patented. It is not possible to patent a new drug in its racemic form today, then expect to market it later as the pure enantiomer, as the issue of stereoisomerism is now an established fact. An example of an established chiral drug is bupivacaine, which is a long-lasting local anesthetic used as a spinal and epidural anesthetic for childbirth and hip replacements. Bupivacaine enantiomers was the first local anesthetic pure-level bupivacaine that was available for clinical use in the end of the last century. Years later, the bupivacaine molecule appeared on the market with pharmacological and pharmacokinetic characteristics similar to those of bupivacaine, but with less neuro and cardiotoxicity. This levo-enantiomer resulted from the separation of racemic bupivacaine. But who grants patents? In the USA, it is the Food and Drug Administration, FDA, and in Europe it is the European Agency for the Evaluation of Medicinal Products, EMEA. This takes the form of an investigational exemption to a new drug application, IND. In the USA, this takes the form of an investigational exemption to a new drug application, IND, which is a confidential document submitted to the FDA. The IND should contain information regarding the chemistry, manufacture, and quality control of the drug, as well as information on its pharmacology, pharmacokinetics, and toxicology. If the clinical trials proceed smoothly, the company applies to the regulatory authority for marketing approval. In the USA, this involves the submission of a new drug application, NDA, to the FDA, and in Europe it's called a Marketing Authorization Application, MAA. After all of this, the manufacturer requires submitting chemistry, manufacturing information, and demonstrating that the product is comparable with the product already approved and the product getting the denomination New Chemical Entity, NCE, or New Molecular Entity, NME, refers to a novel drug structure. FDA monitors the label to ensure that it instructs physicians about the mechanism of action of the drug, the medical situations for which it should be used, and the correct dosing levels and frequency. In addition, clinical investigators ensure that their records are consistent with those provided in the NDA and that patients have been adequately protected. Finally, an approval letter is finally given to the company and the product can be launched. Fast tracking. The regulations of many regulatory bodies include the possibility of fast tracking so that they reach the market as quickly as possible for drugs that show promise for diseases where no current therapy exists, and for drugs that show distinct advantages over existing ones in the treatment of diseases such as cancer. An example of a drug with the fast tracking process is oseltamivir INN, an antiviral drug that slows the spread of influenza virus between cells in the body by stopping the virus and chemically cutting ties with its host cell. Median time to symptom alleviation is reduced by 0.5 to 1 a day. The drug is marketed under the trade name Tamiflu and is taken orally in capsules or as a suspension. Tide is also a drug that the FDA has approved in the condition of fast tracking. It is a neuroprotector for the therapy of paralysis suprarenal progressive. This drug is developed by Nocera. Crizo is an experimental drug for the therapy of pulmonary cancer that the FDA has also approved for fast track. Orphan drug. Orphan drugs are drugs that are effective against relatively rare medical problems. In the USA, an orphan drug is defined as one that is used for less than 200,000 people. Financial and commercial incentives are given to firms in order to encourage the development and marketing of such drugs. The attitude of pharmaceutical companies towards orphan drugs may well change following the therapeutic and financial success of imatinib, an orphan drug which was designed against a specific form of. Since the implementation of EU legislation on orphan drugs 2000 until 2007, there have been 769 applications for appointments to the European Medicines Agency (EMEA). Having reached that status, a total of 499, which means 65% of requests. In total, in a given period, 44 obtained marketing authorizations for orphan drugs. An autoimmune disease like lupus causes the body's immune system to not recognize the difference between healthy and harmful substances, so it begins to attack the organs and tissues of the sufferer. Benlysta is the first drug approved in 2011 by the Food and Drug Administration of the United States in over 56 years to treat lupus disease. The drug was developed by Human Genome Sciences, Inc. Cystinosis is an autosomal recessive inborn error of metabolism in which the transport of cysteine out of lysosomes is abnormal. In the nephropathic form, accumulation of cysteine and formation of crystals damage various organs, especially the kidneys, leading to renal tubular Fanconi syndrome and progressive glomerular failure with end-stage renal failure by the end of the first decade of life. Cystagon is indicated for the treatment of proven nephropathic cystinosis. Cysteamine reduces cysteine accumulation in some cells. Heart disease is a rare neuromuscular disorder that manifests with a broad spectrum of symptoms. All patients typically experience progressive muscle weakness and breathing difficulties. Lysosome was first authorized in 2006 and was the only approved treatment for heart disease at the time. Hans Verder Boy of the Erasmus Medical Center in Rotterdam, Netherlands, a lead author of the study, which was conducted by the biotechnology company Genzyme. Xyrem is a central nervous system depressant used to treat cataplexy, sudden loss of muscle strength, and reduce daytime sleepiness in people with narcolepsy. It is estimated that tens of millions of patients in Europe suffer from some form of orphan disease. As a result, there is a significant market to be tapped. Good laboratory, manufacturing, and clinical practice. Finally, to obtain the evidence needed to show whether a drug is safe and effective, a pharmaceutical company will normally embark on a relatively lengthy process of drug evaluation and testing. The aim of the regulations is to increase the quality of the determinations and tests and to ensure the validity of the results for the establishment of mutually accepted guidelines. Good laboratory practice, GLP, and good manufacturing practice, GMP, are scientific codes of practice for a pharmaceutical companies' laboratories and production plants. They detail the scientific standards that are necessary, and the company must prove to regulatory bodies that it is adhering to these standards. GLP is concerned with the organizational processes and the conditions under which laboratory studies are planned, performed, monitored, reported, and recorded. Regulations apply to the various research laboratories involved in pharmacology, drug metabolism, and toxicology studies. GMP is part of quality assurance, QA, which ensures that products are consistently produced and controlled to the quality standards appropriate to their intended use and as required by marketing authorization or product specification. The GMP regulations apply to the production plant and chemical development laboratories.